Sanger Institute in Cambridge in the UK. The next 3 talks are going to be about genome browsers, so hopefully we'll see some fisticuffs outside about who's the best, so that's going to be exciting. Over to Thomas.
Okay, so yes, it's genome browser time. So this is the visualization session, and hopefully I don't actually need to say that we as humans like looking at things, but if you want a bit of inspiration for just how much we like looking at things, some of the stuff that the National Reconnaissance Office has declassified recently shows just what lengths people will go to look at things like the Soviet moon rocket that I'm showing up there. And biology is no exception. It has always, from the earliest days, been a highly visual science. So 2 different views of the nervous system from rather different eras. And genomics is a much newer subdiscipline, but again, right from the very first days of it, visualization has been vital. We're dealing with a complex landscape in which we can see many different data types, and there are a couple of technologies that both showed up around the time the initial human genome draft was published, and are both pretty ubiquitous. When we publish biology work, we tend to build papers around figures, and I think almost everyone I talk to, when they're publishing something, comes up with figures first and then text afterwards, not the other way around. When we submit something for publication, the figures have to be static. Because allegedly some people still read papers on in-dead tree form, although I'm not sure I've actually met one recently. So the sort of things that might go into a figure is this kind of visualization of a genome-wide association study, and I'm not in the least trying to pick on a particular piece of software here because it's very useful, but it is a very static figure, and in a few minutes I'll be trying to show how you can do the same sort of thing more interactively. So without further ado, I think people doing data visualization should be building interactive tools, and as a starting point, interactivity means things like smooth scrolling, straightforward navigation, being able to highlight which parts of the display you consider to be most important, being able to customize the display, and being able to add things to the display as close to instantaneously as possible and as straightforwardly as possible. So that's an aspect of interactivity. But can we go further than that? And I'm going to argue very much that the answer is yes. So I'm actually leaving the presentation for a moment, but this, as you can see, is the same software now running in a standalone browser window. And we've got here the results of a published association study from the Charge Consortium. And what you're getting from this data are variants arrayed on the genome, and in each case a p-value of how those variants are associated with a phenotype. And right now these minus log p values are pretty low. And another dimension of data we've got layered on here, which may or may not be of interest to some of you, is symbols representing the consequences coming out of a variant effect predictor. So those stars represent possible regulatory SNPs. So it is multi-dimensional data. But how can we actually interact with this? So because it's quantitative, we've set a threshold on this, and that's being displayed by this red line. And I can adjust that, and I can. But now I'm going to actually press this button up here and jump to the next threshold feature anywhere in the genome that is up over the threshold. And indeed, I've got one here. The significance of that is one, and in this sort of genetic study, people tend not to believe single hits. So let's try again. And here we've found something that's far more plausible in that there's a big cluster of hits. Conveniently, this being a cholesterol HDL phenotype, these hits are around a promoter of the CETP cholesterol esterase protein. Is there any more structure we can see in this? Yes, there is, because I can select one variant, make it a reference, and calculate linkage disequilibrium values relative to this. And you see, I'm now fairly close to having reproduced that static figure I had a few minutes ago in an interactive setting. And this is being done with more or less standard Dalliance, no modifications to the core code, a couple of little plugins to do stuff like interacting with a server that's calculating linkage disequilibriums on the fly, plus, and I'll talk about this a little bit later on, a relatively complex style sheet to do stuff like putting stars on the regulatory variants. But nevertheless, and of course this is an interactive tool, and we can pull in a bunch of other data from a bunch of different sources if we want to. So, the applications I have in mind for this is all JavaScript. There's less than 300k of code to use this, and it has an API, so you can embed it in a larger application, or in some other context. So simply you can put these things in a blog post very easily. As you can see, you can put it in your talk fairly easily. One hopes it might be possible to put it in a publication reasonably easly as well, in place of a static figure. And then that's a relatively new thing, but journals seem to be now moving in that direction. And if you can't wait that long, it can also export high quality SVG, which you definitely can put in your paper today. So how does this work? I'm sure a number of technical people have stumbled in here. I don't like servers because, in my experience, servers in bioinformatics tend to give you a Perl stack trace wrapping a MySQL error message. There are plenty of honourable exceptions to that, but in general, biologists don't particularly like building high-quality, robust servers. And when a postdoc does that, he then leaves, and a few months later, the thing has fallen over and isn't running again. So we want to do as much of the work as possible in client-side code that we control. We want to keep the server requirements very, very straightforward. There's just one bit of magic you want, which is a header on your data files, access control allow origin star. that means any web application can access your data. It's a couple of lines of Apache server configuration to do this. You can use an absolutely standard Apache server. I think it's actually a political statement as much as a technical one, that when you're releasing data, not only is that data available to download, but it's available to integrate in other people's web applications. I think it's slightly sad that the history of the web means this is an option rather than the default. But that's the way it is. It's something you need to put on your data. But having done that, Dalliance can fetch data from a bunch of different formats that you're probably using already, like Bigwig and BigBed, genomic data files, BAM for your sequence data, Tabix VCF for your variants. If you've got small data sets, you can just use normal BigBed, Wig and VCF files without any complications at all. In those cases, Dalliance has to fetch the whole file, so don't try and put a score for the whole genome, a score for every base of the genome in a plain text WIG file and expect your computer to survive. But beyond that, they work fine. If you've got a lot of data, a UCSC compatible track hub file, which again I'm sure a number of you are already using, and you can get and that will become a nice little tab of tracks in Dalliance's track selector. If dealing with multiple assemblies, no problem, it can do client-side remapping, and if you're dealing with multiple odd assemblies, it's not too hard to format the alignment data between your assemblies, so you can do your own remapping, and you can add little plugins. You can write complete new backends for this. Once in a while, that's a useful thing to do. Surprisingly often, you can avoid it. An intermediate thing you can sometimes do is write little plugins that filter the data from one data source, or take data from several different sources and integrate that together. And so, for instance, this the genetics track, which I showed a moment ago, is actually a client-side integration of 3 different data sources. So there is a Bigwig file with the p-values for the variants. There's a BigBed file which contains a bunch of other annotation like the variant effect predictor results, and then there is a custom backend which gets the on-the-fly linkage disequilibrium calculations, and then there is a tiny little bit of plugin that merges all those 3 sources of data together to build a single object model, which then gets thrown through the Dalliance render. So compose rather than customize as much as possible. If you want to put your own data on this, either you have very simple server requirements, just an Apache with 2 lines of configuration to make the data accessible. You can put your data in cloud hosting for and again there's an option, at least on Amazon S3, for setting the access control origin headers. Or even though it's a web, you can put password security on, or other sorts of security. Or if you just want to see data that really is very much your own in a hurry, although this is a web application, it is able to access local files. So I'm going to select a BAM file and its accompanying index here. Add that. And we've got a read density view from a BAM file. Zoom in, and you can see that yes, this really is definitely a BAM file. Okay. So, in terms of building non-trivial displays, one of the most complicated things is actually saying how you want data to look. We have a pretty rich set of glyphs and plotting styles. The basic styling system was inherited from the DAS protocol, which some of you may remember, and in fact Biodalliance is still a DAS client, but the style sheet system is probably the biggest remaining DAS influence at this point. So our style sheet language is the DAS style sheet language plus a few extensions to do things like scatter plots, which aren't really being supported by the DAS style sheet. This is something we would like to change, preferably with a style sheet language that isn't XML. But I think we'd actually very much like to consult with other people doing visualization to see if we can come up with some sort of common language for how genomic visualizations should be described. If anyone wants to talk about that in a BOF session, I would be very happy to do so. So future plans, obviously, sort of fill in the remaining formats anyone can think of. We get asked from time to time about better displays for comparative genomics. I did actually get a chance to prototype one of those at the CodeFest yesterday. So we've got two standard Biodalliance instances here, one for human, one for mouse, plus an extra component showing the alignment between the two. And another thing which I haven't shown in a lot of detail is we're starting to come up with interactive configuration as an alternative to handwritten textual style sheets, and I think we want to do more work on that. So thanks to the Tim Hubbard group, formerly at the Sanger Institute where I work, and BBSRC who've been funding some of the recent development work on this. Thank you very much.
Great. So, thank you very much, Thomas. That's a really nice talk. Does anybody have any questions? Probably got time for one question. Oh, is that a question or a wanderer? I've got a question.
Am I the only one? How does it compare to JBrowse?
Okay, that's a kind of predictable question. So there are competitors to this. JBrowse is probably the best known. The two projects were sort of started at fairly similar times. They've had somewhat different development trajectories. They do somewhat similar things with differences. There's a certain amount of code sharing between them. So, for instance, JBrowse uses BAM reading code, which is based on the Biodalliance BAM reading code. I think there's probably potential for more code sharing in the future. I think they're probably both stronger for competition. I hope friendly competition. I would suggest that people should be evaluating both to see which they like the most.
Okay. So, thank you very much, Thomas. Round of applause.